Okay, our last lecture of the semester, we first of all will go over real quick of the virus. Then I'm going to do some of the review. Although the key is already on the eCampus, but something I still wanna mention for the review section as usual. And the first early exam will be afternoon, right after this class 1, 2, 3. I just need a quick lunch, then it's in the lab room. And the second early exam will be tomorrow afternoon from 12:30 to 3:30. We're also in the lab room. Okay, I won't be there. This afternoon, I will be there for 2 hours. So let's talk about virus. Last couple of years, we heard about virus all the time because of the pandemic. So we go over these knowledge will be briefly. We weren't going to spend too much time about that, because more or less you heard about something already. So virus, let's go back to our structure. We talked very earlier. If you talk about virus, it can be as simple as that. So in the middle, you have genetic material, which is DNA or RNA, but never both. And outside, you have a capsid. And the capsid, the major components is protein. So a virus could be as simple as this. Now, the very important thing you have always remember, the major difference between a virus and a bacteria. Virus is a obligate parasite. So what this means, they need a host. That is the key. Regardless of where it is, they need a host. This comes out a very unstudied claim during the pandemic. So they say you ship a box. Okay, this is your box. Let's say you have some foods in there, and that's a ship box. And then they say the virus will be there. And then using X-ray, UV lights, all kinds of things to do the inactivation. Does it necessary? Yes, but the chance is very low because this is a box. That's not a live body. The chance of virus to survive there is very low unless it's already have microorganism or bacteria there already. If it's a pure box surfaces, let's say the benchtop surfaces, they will not have a long-term existing or survive the virus. So that claim makes lots of people nervous. So remember the day you have a box shipping an Amazon bag from somewhere, then you're spending like 2 minutes to spray with chlorine and then putting somewhere to using UV lights, and then they say I want to destroy the virus. You can do that, but it's not necessary because the chance of the virus to contamination on a box surfaces is very low because it's a host. They need a host to do it because it's a parasites. The virus will not exist by itself from nowhere. It's different from the bacteria. Okay, so that's something I always said at the very beginning. That's why our first sentence is important. It's obligate intracellular parasites, which means they survive highly rely on a host, and they will use the host material to finish their metabolic process. And even they lysed sometimes, they will recombine, have a new body using the host cell material. So that's why we have a second sentence said it's incapability of independent metabolism. And virus could be very small. It's only like 20 nanometer or a little large, like 400 nanometer. It's passing through the membrane. So what is that? in the old time, we do not have autoclave. Or in some of the lab environments, we don't have like a heat, UV lights, all kinds of the interventions to kill bacteria. We certainly can use a quick methods. Is using 0.22 micrometer filter system. It is a very simple, a very thin membrane. With this range, most of the bacteria will not pass through. So let's say this is a container, you have water here, and I have a nozzle here which is pumping bacteria solution. If the membrane filtration, the diameter is 0.22 micrometer, the liquid will not passing through. Oh, sorry, the bacteria will not passing through. It's going to be blocked from there. But that's not work for the virus. The virus will passing through because it's small. Okay. And I just said there's only one type of the nucleic acids. It's only DNA or RNA, but never both. So how we classification of the virus? We can call it a DNA virus, RNA virus, or it's a single-stranded or double-stranded. We could be differentiated there based on the capsid morphology. We could even to see it's a naked virus or unnaked virus. Depends whether they are the packaging, and the packaging called envelope. And we also could say it's a human virus, animal virus, or plant virus based on the host range. So that's a classification. Now this is what I just draw in there. You will see right here the left side, this is the unenveloped virus. So we have a capsid, we have a nucleic acids. And right side, that is more completed construction of a virus, which is an enveloped virus. So you see the spike capsid, nucleic acids, and the envelope outside. Now, remember the day we talked about coronavirus, that's what it looks like. It's like this. Then outside, you have a package. But so what the package looks like? The package looks like a crown, like that. If you remember the day we had those fact sheets all the time from everywhere, from CDC, that one looks like this. And the outside looks like a crown. So, when we get the vaccine, we actually got from the protein right here. This is a capsid, and we get some protein from here. We also get the protein from here, which is S protein, from the RNA, and then we purify, become a vaccine. So that's what the coronavirus, is a very simple picture looks like. Now for all these spikes here, this is easily to be doing hemoglobin, which is integrated with the human or animal, the blood cell tissue, and then could be penetrate and cause all the problem. And at the beginning they will form, which is indosome at the low pH inside. So that's very similar to what we talk about the AB toxin. This is a different picture. You can see what it looks like. Different morphology. You have a herpes virus, Vaccinia virus, rotavirus. This is bacteriophage. We're going to talk a little bit more about that. And the mini virus is very large, although we say it's a mini tobacco mosaic virus that studied a lot for the morphology and all the others. Okay, we're going to talk about the phage separately. So we talk about the capsid. The capsids we already say this is a protein. So basically it's a protein. It's to protect the genetic material and help to transferring between the host cells. Now, the capsids also have a units. They are composed by, these are very simple units. Very look like a flower piece. We give them a name, we call them protomer. So that's a single unit of the capsid. Under the capsid, we have a different morphology. It could be helical, icosahedral, or complexed. So based on the morphology of the capsids, we can say it's a helical virus. We can say it's an icosahedral virus. That's based on the classification of the morphology. So here you will see it's a helical capsids. A very good example here, which is a self-assembling protomer, and looks like a skirt. The girls' skirts wear during the dancing. This is a typical picture for tobacco mosaic virus. And the nucleic acid is inside, which is RNA, and it's very, very long. It could be sometimes longer than the 10 meters could be. So that's when people get isolated, highly twisted and folded. And you also can see some of these icosahedral capsid. Now, it could be 20 equilateral faces or 12 verticals. And then based on these capsimers units, it could be pentamers or hexamers. So the most widely existing is 5 subunits or 6 subunits. Now the next one, what we want to talk about, this is a complex capsid. And this is a very typical example is for bacteriophage. So bacteriophage, we took this out. You will see it's very similar. It's like this. You have a base right here. Because this base is for them to penetrate, to attach to a bacterial cell. Then, the genome from the center, they could be go inside of a bacterial cell. And they could be just free swimming there, you can see. And they also could be find a genome of the bacteria and integrated there, become part of the bacterial genome. And this is usually get to trouble because later on this phage become a toxin. That's what we call it a lysogenic conversion. Now the phage, they could kill a bacteria, which is called lytic. Or we call it a warm, friendly phage. They could be stay with the bacteria the most of the time. We call it a temperate phage. So that's just a very brief picture, but we're going to talk a little more. So genome structure. In the virus, we normally see all kinds of the structure of the DNA or RNA. So that could be single-stranded or could be double-stranded for both. The genome size is varied between different viruses. It could be as small as 4 or could be several hundred. And they give you an example like influenza virus have a segment genome composed of 8 separated pieces of RNA. Now, during their life cycle, they have a very unique situation which is called eclipse phase. So, which means at the initial of their infection of the host, they are not observed, but they started to working there and later on become infectious particle. and they will reappear in the cell, which means some of the virus caused the disease at a very early stage. They are hiding somewhere. It's not easy to be detected, but it's still apparable, although we do not observable. This is very similar. What we call talking about is viable but non-culturable. But the difference from the bacteria is that they later on will be reappear in the cell. The host specificity, what this means, bacteria can go everywhere. Virus usually have their specific host. So let's say the rabies, we call it a crazy dog virus, because most of the time the dog is a host. But something related to dog or the other mammals like skunks, humans, and the bats could also be their host, but they are in the same category. But rabies will not happen on the plants. So that's the difference. We're also going to talk about like a phage. So we mentioned this phage a little bit. Phage in the clinical area, we talk about lysogenic conversion, which is how they cause the disease, bring the toxin. But in the food safety area, we can using bacteria phage to kill bacteria. So it's another way to inhibit the bacterial growth. They already have a commercial products, which is like Listeria phage, is commercially available to be used. However, they do have a restriction, is they are very specific for the strains. If the phage works for Listeria, they may not work for E. coli 0157H7. Even lots of the time, it depends on different species. The different genome type even have a specific phage related to that. So use the phage have a very limited condition, is you have to find the target bacteria. And also most of the time, the bacteria has to be already grown, has a certain population. So let's say in meat products, you already have a bacteria about 10 to the power of 3 to 5. Then you spray the phage, that might be works because the host is already grown. But if there is only like 10 cells there, it's not going to work because the host number is not enough. Another thing is that usually the phage work more effectively in the low temperature, in the refrigerated temperature, like 4 degrees Celsius, because that temperature will let the bacteria doesn't grow dramatically, and somehow the phage could be attacking the bacteria. So there are lots of the limitations to using bacterial phage as a biological intervention to killing the foodborne pathogens. But they do have a lot of research has been going there. This is what we're meaning for phage are specific for their bacterial host. This is the slides. You should mark a star there. This will be one of your final exam question. How the virus go into a host cell. So we already talked about they need a host, how they go. First of all, they attach to the host cell. Then it's an entry. Then you uncoding of the genome. The genome of the virus will be released, like here. And then they use the host cell machinery or the material to biosynthesis their own genome. It's the same as we talked about for the DNA replication, transcription, and translation. Then they do assembly. That's also in the host cell. They're using host material to do the assembly, and finally will be released. Now this is usually because of lytic cycle. Lots of the times, often they also integrated into the host genome. So attachment, entry, uncoding of the genome, synthesis, assembly, and release, those 6 dots will be the key of the steps to answering the final exam questions. It's already in your practice exam questions. So attachment. So how they attach? They depends on it's enveloped or unenveloped. It is non-enveloped virus, most likely it's a capsid, it's enveloped virus, it's a protein in the membrane. Now, all the attachments, they have a specific site for that. It could be a membrane protein, it could be polysaccharides, it could be lipids. This is something I want to mention, is that we talk about the virus have a membrane system. Then they go to the host membrane system. They going through the process most of the time. We call it a fuse. And the fuse, they will generate a very special structure. It's called endosome. So this is also endosome. That's why the process we call the endocytosis. So let's say you have a bacteria inside. And this inside, usually the pH is lower, it's around 4 to 5. They're very similar to the AB toxin. Remember the AB toxin have an endosome for Clostridium, for the Corynebacterial diphtheria. The AB subunits in the release, it's very similar to the virus here. So we had a picture you can see. They go inside, they first fuse together, and then they release to the inside, and they have these spikes, they turn away, and then these environments, which is low acid, and then they release. Now these 2 pictures usually is for enveloped virus. If it's naked virus, they're gonna come in, going through the face, have an endocytosis and directly release the genome, go to the host cell. Okay, they do not forming the endosome, the low pH endosome. This is the synthesis. Like we just said, they use cells, enzymes, and monomers. They're also going through replication, transcription, translation. And now they composed by their own protein, it's already being programmed. So it's step by step. Some of them, it's a major important function. Or it's more important, we call it early proteins. Those first will be interrupted the host cell normal function. And then those structures, like capsid, protomers, or other structured proteins, they will synthesize later. So that's how they create it. So you can see the base plate protein going through this way. Have a teeth and a tube. The head protein going through this way, have a prohead generated. And they have almost finished the bacteria, this bacterial virus. This is a phage. Then the tail and the fiber protein go there and finally become a bacteriophage. So the head is more important, and the teeth is also important to hold on the head. These 2 things we're going to do first. That's early protein, and then the tail, fiber, and the coral, that's later. So the finally synthesized become a new virus. This is very easily to understanding. In our first lab, we talk about the fungi. It's very easy for that. Virus into the host cell, going hemoglobin or hemagglutinin. They will have lots of spikes. This is happening in the host cell most of the time is splitting. And when they go inside, they form this endosome, which is connected to cell membrane system. It's very similar to, we talked already about the fungi body. So how they generate a daughter cell like a fungi. And then after budding, this enveloped virus is going to the host cell. So this slide could be as wordy as it is, but the keywords here is the budding virus. That's important, the key function of the viral release process. Okay, bacteriophage. Bacteriophage, they could be going through lytic cycle or lysogeny cycle. So we already talked about that. Lytic usually causes lysis of host bacterial cell, and lysogeny usually the virus DNA or RNA sequence is integrated into host bacterial cell. This usually becomes a toxin. That's why we call lysogeny conversion, the process. This is important you understand how the toxin will be there. This is already talked. It depends the phage's growth situation. They could be temperate, they could be lytic. It really depends on the environments. Sometimes you have a stress environment, they will become a lytic. In a moderate or friendly environments, they become lysogenic. In the lab, we also use them, like we just said. It could be used to kill foodborne pathogen if it's a lytic phage. But it also could be a lysogeny phage, which is they integrate the genome into the host cell. So there is something interesting. So let's say I have a beef. Okay, contaminated with E. coli, 0157H7. So traditionally what we do, we're using lactic acid, for example. Lactic acid is antimicrobials. We're using 2.5%. How many we can kill? We can kill one log of bacteria. So some people will say it's not enough. Or let's using some creative methods. So we're using E. coli 0157 cultivated bacteriophage. Now, there is another advantage for that, is if we are using lactic acid way too much, these bacteria could become lactic acid resistant. This is what you did in your genetics puzzle all the time. So, people are saying, how about I use bacteriophage? I could solve the problem for lactic acid resistant. But sometimes it works, sometimes it's not working. The reason is, it depends on bacteriophage. If the bacteria in here already have this chunk of sequence, which is LAR, and then once I have a bacteriophage goes there, this same sequence, because like I just said, even the lytic phage could also be lysogenic, this could go here. Even like 5% of the bacteriophage become a temperate. This phage could carry on this resistant gene. And then if this phage is intaken by another bacteria, it could naturally transfer there. So, in the early time, people think one of the way to prevent antibiotic resistance is to developing bacteriophage. We call it a phage therapeutic. But when we do, more and more research has been showing it works sometimes, but sometimes it doesn't work, because even if you have a 5% of the bacteriophage during their lytic cycle, they are, instead of go lytic cycle, they go to the template, which we call it a lysogyny cycle. They could carry on the gene, and if this goes to the environment, they will transfer to another bacteria. And we have a terminology already called transduction. Remember transformation, transduction, and conjugation? That are the three ways for bacterial genetic information transfer. The transduction, they need a bacteriophage. So that's something I want to mention. Lots of the research has been going on there in the last 10 years. Sometimes it's failed. But one of the reasons is for the restriction of the bacteriophage to using. Another one is even a lytic phage, they have a certain percentage of the chance it will become a temperate phage. So this is what happened. We have a lytic cycle, we have a lysogyny cycle. If lytic cycle, certainly we call the burst of the bacterial cell, which is lysis. Then the virus using the host machinery to have their own virus. Lysogyny cycle, instead of the lytic or lysis of the bacterial cell, their DNA or all RNA is integrated in there. So that piece sequencing most of the time is a toxin. That's why every single passage we could using a theory called lysogeny conversion to explain that. Which means long time ago, there is a virus, there is a bacteria. And for somehow the reason, the virus attacking the bacteria. Instead of lytic of the bacteria, the virus genome integrates into the bacterial chromosome and stays there forever. And with a lot of the generation development, the phage become a toxin. That let the bacteria very pathogenic. So we mentioned Corynebacterial diphtheria is caused by the bacteriophage, the beta phage. We talk about the Staphylococcus aureus toxic shock syndrome, that is also coming from a phage. And the botulism coming from a botulism toxin phage, Chagas toxin, that's for O157H7, and chlorotoxin, that's all coming from a phage. It's just one of the theory, that's called the lysogeny conversion. It is very similar to another theory we talked earlier about the endocytosis. So why eukaryotes has a very, you say, complicated cell machinery? Because a long time ago they think those are bacteria, and the bacteria, or those are prokaryotes, and then they go to a eukaryotes and stay there, and develop to become a cell machinery. That's the same theory about that. Okay, how to cultivation detection viruses? Then you always remember, they need a host. So, this is very interesting. Lots of the time, we had agar plates, and we can use to cultivation bacteria. And then, surprisingly, we can also cultivation for virus. Then lots of people will say, you just said the host. How come the agar can cultivation virus? Because we omit one important factor. If an agar can be used to cultivation virus, they must first have a layer of bacteria. Without this layer, virus will not survive. This is the key. You always remember. So the bacteria is there. And then I pour prepare the virus solution, you will see lots of these dots. It is transparent. Like beta hemolytic, it is transparent. Like that. That is called plate. So they give you a wrong impression. Agar can cultivation bacteria. No, they can, but you must have a bacterial layer there. Now, obviously, those type of virus is bacteriophage. Okay, how we going to cultivation or quantify a bacteria? Remember, we have a colony forming units. So for phage, we have a plague forming units. So instead of CFU, we have a PFU. But the acceptable zone is different. PFU is 20 to 200. CFU is 30 to 300. The reason is PFU is sometimes difficult to see. So we need to let the standards a little bit different. Okay, a range a little bit smaller. So that sometimes we can do. But most of the time, we're using chicken embryos, eggs, to cultivation the virus. So that's how we do is a plague number. We do PFU. Other way we can measurement to the virus is called LSD50. The dose of the virus you injected into an animal or a group of the experimental animal cause statistically 50% of them are dead. That is called LD50. We're also using LD50 value to evaluate the amount of the virus. Most of the time, this index is using in the pharmacy. In the pharmacy, animal experiments to evaluate the function of the medicine. But keep in mind, this is LD50 is statistically 50%. Does not mean you 10 rabbits there, you're going to have 5 killed. It could be sometimes 6, could be sometimes 3. They have a model and a formula to calculate it, not exactly the middle half of that. Although we call it 50% death rate. Okay. Something else, veloids, that is composed by circular single-stranded RNA. It's a plant disease. Let's say 1996 in Philippines, there is outbreaks for that. All the palm trees has been damaged. That's veloids. In the first class, we talk about that. Another very magic stuff could be. So we talk about that. That's a virus. If I don't have this, what I left is a protein. Is protein could it be a microorganism? Yes. It's called prions. The prion will cause a severe damage of the brain. It is as simple as just a protein. So scrapie in sheep, medical disease in cows, Kulu and CJD, which is in the humans. So what do you know about the Kulu? You keep laughing. The muscle is always relaxing, it's never being contracted. So think about if you laugh 4 hours, what it looks like. So that's the end of the class. So we finish about here. Most of the time we focus on the bacteria, but we mention a little bit about the virus at the end. Okay, let's go back to see what we're going to do for the final exam. First of all, there is a question there. Talk about how we develop the active immune system. I already put, wait a second. Why can't I download it? A picture there. So what happened is very ironic. This is the picture I really like. It is existing in a book about 20 years ago, when I first started to teach. And after the new version comes from the McGraw-Hill textbooks, they never have this good picture. But it's really very nice and understandable what happened. For the applied development of the human immune system, or we could say acquired immune system development, it's really nice. You have a B cell line, too large. B cell line right on the top. Right on the top, bone marrow goes to lymph nodes and the spleen. T cell line, the thymus gland, go to the lymph node and the spleen. Depends on what type of the antigen it is. It's a free soluble antigen, or it's a complex antigen. We go to B cell line, the T cell line. It's a really nice picture. This is a very good explanation for the question in the final exam. So that's why I have this specific figure to you. I want to put them into the slides, but the slides never have that picture anymore. So I don't know why they took this out, and it's really very good picture. So just want to mention this will be the major question in the final exam. So let's talk about these things. I don't go over all these details because we have a time limited, but I want to make sure several big things you need to know. Because in my key I already put the answer there. What is the major thing we're going to do? Is right on the top. Those are 5 questions. That's you need to know, because it's very important for your future life or find a job. So first of all, you need to know those acronyms of those major agencies and the major antimicrobial steps happen for the applied pathogen control process. So, very simple. What is the FSIS? Food safety, inspection, service. So that's very interesting. The current director, Justin Rasson, is one of my friends. When we were at Colorado State University about 15 years ago. So he was graduated about 3 years ahead of me. Then worked at McDonald's, OSI, some food companies. And this year he was being nominated by the Secretary of the Agriculture, and right now is FSIS director. So that's why those agencies are important for the food safety, FSIS. So what they are doing? Poultry, meat, and processed egg, is that right? Raw egg or processed egg? Processed egg. So what is FDA? Food and Drug Administration. So in the food safety area, therefore, the food products excluded poultry, meat, and raw egg. And the raw egg is belonging to that. Okay, what is CDC? Centers for Disease Control and Prevention. Don't forget about this. Don't forget about prevention. That's why the vaccine is coming from there. Okay, that's the three major agents. What is gap? Good agricultural practices. What are the major things we're going to do? Clean hands, clean surface, safe soil. We don't say clean soil. And clean water. Remember zero tolerance for E. coli, generic E. coli, and fecal coliform. Okay. What is FSMA? For the safety, Modernization Act. When did it become a law? Does that mean every fresh produce grower needs to be fit to FSMA? They give very small growers flexible. Okay, that's FSMA. Okay, what else we have? What is we called? What do we call this? Inside, outside, birds wash. Okay, what are the major antimicrobials we're using? That's perioxycitic acid, PAA, and chlorine. So we have a question for that, okay? This, we have another acronym, is online reprocessing. Okay, ORL. So these are the acronyms, you should be knowing this. Now what we left, a major one. What it has on? This will be somehow has those certification programs or certification classes. If you're interested about food safety, you should get those certification. So I mentioned like Pennsylvania State University and State College, about like 3 hours away from here, they have the training all the times. So what is HACCP? Hazard analysis and critical control points. Okay, so what did it talk about? We had the 7 principles. Conduct a hazard analysis. Second, establish a critical control points. We call CCPS. Number 3, establish a critical limits. This critical limits is a parameter, which is a physical or chemical parameter corresponding to the critical control points. But we said this never should be a range. It has to be a fixed number. In HACCP, under FSMA, you can have a range, but it's different. So here we had a HACCP. In FSMA, we had preventive control of the human consumption foods. It's very similar, but if we talk about HACCP, this has to be a fixed number. Okay, establish critical limits. Establish monitoring procedures. Establish and conduct corrective actions if affiliation happens. Conduct validation and verification procedure. Last, record keeping and documentation. Can you do that? That's very important for you to know the HACCP, especially in this area. Even you go Pennsylvania, we talk about the meat poultry processors. In the future, if you have an extension specialist job, we got to know to all these things. Now, every food product is different. That is something further on. Lots of the university have a specific class talk about HACCP. We don't have it here. You certainly can get certification in some places. Okay, so that's something we want to emphasize. Another question what we want to talk, what is the temperature control important during the scottering? Because that class I didn't teach because of the weather. But I want to go back to talk about this. We'll emphasize this because it's a very interesting topic. We should talk. Not this one. Exam 2, the slides. I will have a question to ask you about that. Because lots of people doesn't realize about it. Okay, we go here to the scottering. It's right there. So there's the hard scottering and the soft scottering. The temperature range is important. However, we have a misconception. So first misconception is people say you use hot scottering is 64 degrees Celsius. How about I use 70 degrees Celsius? So I can kill everything. Because it's a very, very hot temperature. It can be effective to kill bacteria. But what happens? If you're using 70 degrees Celsius, the surface of the chicken looks like lipids. So if you know about this, two, or three, this usually unsaturated fatty acids. Unsaturated fatty acids very easy to be extracted. We call it lipidation. So looks like you're not scottering, you're extracting lipids. Then what happens? This will happen. The fatty acids will be accumulated if you put in the chilling tank. Look like a dust. And they will be prevent the working of antimicrobials. So you see 40% of the positive sample in come out 53%. Not only not decreasing, the pathogen is increased because of the cross-contamination. So that's important. Now the second thing. People say, how about I use 45 degrees Celsius? Warm water. Because I can easily to handle and to do the defeathering. If you do 45 degrees Celsius, we call it's very expensive incubator. Why? Salmonella we call mesophile. What is the arrangement of the temperature for them to grow? That's very close to that. So at 45 degrees Celsius, you may kill a mild amount of the salmonella, but let them rapidly grow. So if you are using 45 degrees Celsius to do the scattering, instead of your scattering, your cultivation bacteria, your growing bacteria. So the temperature control is important right here. That's the reason. Now others, of course, is including the water has to be countercurrent, and do not use oxidants because off-gas will generate. So that's why scattering is important. Lots of people don't know about that. You can see lots of people using 40 degrees Celsius just defeathering then your cultivation. And later on, you can even smell about it. That's why you have the chicken flavor, because the lipids comes out sometimes using very high temperature. Okay. I don't think anything else we need to talk because I already put the key there. That's all we have. One more thing, maybe. Okay, agglutination and opsonization. That's a question is important. If you didn't know, not the video, if you didn't know about that, here I have the slides. The slides right here, we talk about, that is the key. It's very clear what is opsonization. So they interacted with each other. And in the FC segment, we call the crystallized fragments of the antibody. They bring the macrophage. The macrophage goes there, and easily to be break down the antigen-antibody reaction. That's called opsonization. Now, what is agglutination? Instead of you have a simply precipitation, they connected with each other and becomes a clumping. So agglutination is actually could be using to testing salmonella or E. coli 0157H7, those testing kits. Okay? So that's a picture is important for you to understand these two important terminology. Now lysozyme we talk about. The phagocytosis, what they do? They break down the glycosidic bonds of an acetylglucosamine and an acetylmuramic acids. That's for lysozyme. So, Helicobacter pylori, we already mentioned in the lab already, having ureas. So, that's all we have. Okay, last thing. I want to make sure we are on the same page. Bonus points, 30 points, but you need to handwriting in the classroom. It's not you already typed pages. You can prepare, but in the classroom, you need to use handwriting to write 250 to 350 words. And make sure your handwriting, I can recognize them. And the key point here is that I will counting the words. And do you have an important scientific terminology there? You cannot like writing a novel or something. But of course, you need to share your own thoughts. Now, I want to say this is a debate question. There is no white and black answer about that, as long as you justify. Okay? If you want to do, you need to write in the classroom. You need to spend time there. If you already being happy about your final exams, you don't have to do. Because when you do, I have to grade it. It's spending a lot of time to do grading. Okay? So what I do is, I graded all your normal final exam, plus the 5 points of attending Dr. Scott Baldridge's lecture. If you are already above 360, then I don't even see this. If not, I'll go back to grade the essay extra points. And we are trying to see whether you reach 360 or 340. Okay, that 340 is A minus. So just want to let you know. Okay, last thing. This is what happened before. In the final exam, some of you may only need 50 points to get an A. So what do? You study only that 50 points and left the whole exam paper empty. And there is no chance to do that afterwards. It's done, it's done. So, what is my suggestion? Just take it as your normal exam. Study slides, pay more attention to the practice exam questions. The key is there. And don't do it very rough. Okay? Because if your exam paper is empty, then no way we can help. So, make sure you study normally and do not take the chance. Even you only need 50 points, you study normally, you get 80, you get 70, that's okay. Then you can have a good Christmas. Okay, so that's something I want to say. Is bonus points, make sure your writing, have a good handwriting, and just to do a normal study by yourself. Don't take the chance. So, assuming you don't know what you did for the exam 1, 2, 3, just try your best to do the final exam. Or keep your normal pace, what you did. Obviously, if you're not very well for the first 3 exams, you need to study harder for the final. So I can tell you, we had a very divided classroom. So even the lab final, we have people get 81, we have people only get 12. And I have a couple only get like 20 or 30. So just want to let you know that you still have to work hard. There is at least 140 points is up in the air for grab. So it's not the time to give up. And also not the time to do it roughly. Okay? So that's all my suggestions. Done.